I'm Jennifer Shoesmith and I'm a PhD student at the University of Dundee. I work in Sarah McKim's lab at the James Hutton Institute as part of the Division of Plant Sciences. I use barley as a model organism, which is a cereal crop similar to wheat. Barley is used to make beer and whiskey and is also used as an animal feed. It is a good model organism for cereal crops as it has only 2 copies of every chromosome, like humans, which is simpler than the 6 chromosomes found in wheat. We also have a lot of advanced genetic resources available, such as a new genome sequence. Also, in comparison to some other cereals, barley has a relatively short life cycle, which helps us to do more experiments faster. Barley also grows in a wider range of climates, which makes it an ideal crop to study adaptation and to learn how to improve crop productivity or resiliency under environmental stress. In my PhD, I use dissection microscopes to do fine sampling of plant material. I also use scanning electron microscopes to image grass petals to see them in more detail. Grass petals are different from typical flower petals and form small, fluid-filled, fringed sacs which we call lodicules. When grass plants flower, lodicules expand to separate the outer protective floral organs to open the flower. This allows pollen to escape the grass flower and fertilise other individuals. As part of my PhD project, I have been imaging the lodicules of mutants which have defects in a developmental gene. In Bowman, a variety of barley, the lodicules expand to open the flowers. In a mutant which expresses more of our gene of interest, the lodicules are much smaller, which means the flowers remain closed. So, this mutant can only self-pollinate. In another mutant that lacks our gene of interest, the lodicules are much larger, with extra tissue. This leads to extreme open flowering and potentially increasing pollen transfer. Studying the genes controlling lodicule growth is important to help control pollen exchange in barley, a desirable ability for breeders. For instance, varieties that only self-pollinate may be favourable to prevent the contamination of hybrid varieties and to stop the release of transgenes, whereas at other times, a crop breeder may want to promote pollen exchange between different barley varieties.